I don't think you need much of an introduction. I think the number of people that I'm still admitting from the waiting room speaks to that. But I think it's a really interesting point in time in the program that you're presenting, because you have pretty much popped up in every previous presentation that was leading up to this so far. So I think people have a good primer for what to expect from you. I will share more bio about you in the chat and a little bit about your ongoing projects. I think last time you presented here with Vadim, which was, I think, entirely off the record. Let's see where we take the discussion today. I'm really happy and totally honored that you made time for this. And I'm super excited for the conversation, and I'll be in the chat. All right, the stage is yours, and I'm making you co-host.
All right. Many of you know all about methylation clocks. Today I want to talk about what's new and what's missing. You know, I will, and some philosophical debates about the aging field. So I will talk about clocks, and just to be very precise, for me, a clock is fundamentally a procedure for arriving at a number, big emphasis on number, you know. I mention it because the word biologic clock or molecular clock also has many other definitions. Some people think of a clock as a causal molecular process that underlies aging, or a set of genes, or a set of proteins, or special cells. But in this talk, it's all about an algorithm, a number, a biomarker. And let me already come to one topic for future human biomarker studies. And as many of you know, I developed a so-called pan-tissue clock that applies to all human cells and tissues. But the question is blood a good surrogate tissue for other organs, right? Is it sufficient to simply measure blood methylation? And to get at that, I generated here methylation data from roughly 120 in post-mortem samples, individuals for whom we had measured, you see on the diagonal, blood, bone marrow, spleen, lung, muscle, adipose, kidney, heart, liver, and so on, right? And then you can define this measure of epigenetic age acceleration, right, as a residual of methylation age, where you regress out chronologic age. And you can ask, what's the correlation of epigenetic age acceleration in blood versus bone marrow, as an example? And it's a high correlation, 0.71, or blood versus epigenetic age acceleration in the spleen, again, pretty high, 0.74. But then you see a surprising result, which is liver. Do you see the correlation between blood methylation, age acceleration, and liver age acceleration is negligible, you know? And this kind of analysis makes the point that the current clocks, yes, I mean, you can look at it as half full or half empty, you know. The current clocks show pretty remarkable conservation across tissue types, or you can look at this as a dismal failure and say, well, maybe blood age acceleration is not really related to heart age acceleration or liver, you know. And so I feel there's an opportunity, you know, to develop new human biomarkers that really hone in on cytosines that are highly conserved, whose aging effects are highly conserved across tissues, so that a blood sample, or let it be a buccal sample, or a buccal swab, or a saliva samples, or an adipose samples, you know, it would be nice to have one source of DNA where you can capture the epigenetic age of other organs. Extreme example is brain, right? We all want to measure brain aging, but what accessible tissue can serve as surrogate? I wanted to make this point. I show you here as a finding that was surprising to me. The upper row shows the effect of hypertension. This is now the same study. And what you see is in panel A, we see that hypertension was associated with age acceleration in adipose and kidney and liver and muscle. Interesting, you know, but not all organs, you know. So the other conditions here make similar points. So it could be that certain age-related conditions affect epigenetic aging in many organs, but others will only have a very precise effect on one organ, you know. So this is under the heading, again, is let's say you want to predict morbidity. Is blood methylation enough, or should you measure methylation in multiple tissues and cell types? You know? Moving on to another topic, because today is all about discussion, you know, so I want to go through some slides so that we can debate things, but this is under the heading new developments. So as many of you may know, we developed a new Infinium array for measuring methylation in all mammalian species. This mammalian array can be purchased through my nonprofit, the EpigeneticClocks Development Foundation. You all know perhaps Bobby Brook, and so I just want to encourage you to reach out. It's a non-profit, so we try to promote the use. The array was designed to tolerate mutations across species. So here in the first row, you see a sequence of nucleotides, and what you really want to measure is the cytosine at the end, right? And as you look at other species, you will observe that some of these nucleotides are mutated, you know, but you still want to measure this one cytosine at the end. So what do you need to do? You need to design oligonucleotide probes that still bind, you know, and that's very much done by this mammalian array. So it's designed to tolerate mutations. And here's a random application. We built very accurate clocks for the naked mole rat, an animal that has negligible senescence, but according to EpigeneticClocks, it definitely ages. Another application, more exciting application. This is now a dual species clock. So this is one mathematical formula, more precisely one regression model that measures relative age in both humans and rats. And the lower panel shows you orange dots that correspond to rat samples, and green dots are human samples. And you see they overlap and overlay. Why? Because carefully look at the x-axis. The x-axis is a number between 0 and 1. Why? Because we defined this measure of relative age, which is defined as chronologic age in units of years divided by the maximum lifespan of the species. So by definition, this ratio will always lie between 0 and 1. And it's just one of these miracles of cytosine methylation that you can build very accurate dual species clocks. Now, we didn't stop there, and of course aim to develop what we call right now universal mammalian clock, but in hindsight, a better name is pan-mammalian clock. So these are now EpigeneticClocks, one regression model for measuring relative age, for example, in all mammalian species. And here I show you some results from an application over 10,000 tissue samples. These samples are labeled by species. We have here over 150 species. And the upper left panel shows you chronologic age versus the methylation age estimate. But the lower panel is kind of the real meat, because these clocks estimate relative age. Again, the x-axis shows you relative age for each species separately. And you see the methylation levels predict relative age with a correlation close to 0.95. And it's a miracle that this is possible. Why? Think about, we're talking maximum, we use maximum lifespan, right, in the denominator of the ratio. And often the maximum lifespan of a species can only be estimated. Think of the debate of human maximum lifespan, you know, but in many species, maximum lifespan can only be approximated. So even though there's this substantial noise in the dependent variable, the outcome, methylation still allows you to get this very accurate correlation. And The middle panel shows you the application to marsupials. In other words, kangaroos, opossum, Tasmanian devil, and so on. Why do we show it? Because these marsupials diverged from eutherians, and the mammalian array only covers only half of the cytosines on the mammalian array even apply to marsupials, you know. So it's really reassuring that in this worst case scenario, application to marsupials, these universal clocks still work. Want to mention, we have also another universal clock that does not make use of maximum lifespan. Why? Because maximum lifespan, again, is a noisy characteristic of species. However, people have a pretty good estimate of age of sexual maturity of different species. And so this, what we call clock number 3, is a universal clock, but it makes use of age at sexual maturity to calibrate the ages. Here I show you how the universal clocks work in specific species. The upper left are humans. I mean, the universal clock clearly works very well in humans. Upper middle panel is mice. But you can see these clocks work really well in most species. Different topic, forget chronologic age. Now we want to come to a fundamental life history trait, which is maximum lifespan. Human maximum lifespan is assumed to be 122. The rat has a maximum lifespan 3.8 years, and so on. Question, can you predict maximum lifespan based on methylation measured on the mammalian array? And I think the answer is clear. These are so-called leave one species out analyses, and you see a correlation of almost 0.9, you know. So this is one of these profound insights that methylation allows you to measure these life history traits. Now, we have this estimate of maximum lifespan, and question, what about growth hormone knockout mice? You know, these growth hormone knockout mice are dwarf mice, right? And their lifespan is very much enhanced compared to regular sized mice. And interestingly, the left panel shows you that this maximum lifespan predictor shows the expected association. You know, the knockout mice have a predicted maximum lifespan that's older than the control mice. You know. The right panel shows you caloric restriction, and there is no effect, you know, which you can think of as either disappointing or we can discuss it. But everybody knows caloric restriction enhances certainly average lifespan of a mouse, right? No question. But the question is, does it really change the species characteristic, you know? And according to this analysis, it does not, you know. So maybe we talk to the mouse people later, but it's very important to distinguish a species characteristic such as maximum lifespan from what people care about often in the aging field, which is a risk estimate of mortality risk or morbidity risk, right? Because a lot of things relate to mortality risk, but they may not change the maximum lifespan of the species, you know. I would say caloric restriction in humans, right? Do you really change the maximum lifespan of our species if you starve people? I'm not sure. Different topics. Are we ready to wage war against aging? And here I use this quote from Donald Rumsfeld: you go to war with the army you have, not the army you might want or wish to have at a later time. This is always the quote that I think of when we hear speakers from the FDA and we talk biomarkers, right? Yes, in 20 years, we will have amazing biomarkers. The problem is we need solutions right now. Do we have biomarkers that can be deployed today, you know? And my answer is kind of, you know. So many of you have heard me talk about this GRIM age biomarker. It's our best estimator of mortality risk. I'm quite optimistic about it. I'm aware of its many limitations more than anyone else. However, it does have strengths. And one is that there's really very strong literature support that GRIM age predicts mortality risk and a lot of age-related conditions in cross-sectional studies by independent groups. Also, people have started to conduct these longitudinal studies where we relate rate of change in GRIM age to increased hazard of death. And they look good to me. From what I've seen, they are encouraging results. And why do I mention longitudinal studies? Well, this is under the heading causality. I also want to mention large genetic studies. So we analyzed SNP marker data and methylation data from 41,000 people, and we developed so-called polygenic risk scores for GRIM age. And these polygenic risk scores were associated with parental longevity. So why do I mention it? Because epidemiologists often use genetic arguments to argue causality. And then another point 4 is more mundane point, but it's important. What is known as analytical validation. You want low test retest variability. So GRIM age performs exceptionally well. And now I would, coming back to Rumsfeld, this is what we have right now, but I'm very excited that many groups, including my group, we are continuing to develop new versions of these biomarkers, So the longer you wait, the better the version. I often use the analogy of the iPhone. There's iPhone version 1 and now there's iPhone, I think it's iPhone version 12, right? Things get better, continually get better, but we already have things we can work with. I now want to talk about challenges in the aging field and more particularly in the biomarker field. These are more philosophical discussions. Hopefully you don't mind, but I want to start with the fundamental challenge of biologic age. The problem is that mathematically speaking, it's a latent variable. We don't have a definition of it. Latent means you need to infer it. And inferring it from quote unquote manifest variables, mean actual variables. So for example, what my group and many others have worked on are using omics data, right? We have methylome, epigenome, transcriptome, proteome, metabolome. And so you can use these molecular readouts to try to infer biologic age. And conversely, a pathologist would look at cellular changes, tissue pathology. And then there are, of course, all these clinical readouts on the right-hand side, anything from frailty measures and so on. And the field, our field of aging biomarkers needs to find a way to integrate this information. Now, we have one good starting point, which is chronologic age, because the bottom line is nobody ever debates that biologic age increases with chronologic age, so we have a starting point. What does it mean? If you have a biomarker of aging and it does not correlate with chronologic age, you have a problem, It may measure very well mortality risk, Take viral load. You take some infectious disease, it may strongly relate to mortality risk, but it may not at all relate to chronologic age. Suddenly you have a problem, Personally, I'm interested in biomarkers that measure what I sometimes call innate aging processes, and all of them have a strong correlation with age. And here is a challenge in the field, the aging field. there's really no consensus on an established theory of aging. And why do I say that? Because we may very well have already a wonderful theory of aging. I'm sure we could spend 2 days describing very plausible aging theories. But I would say it's fair to say there's no consensus. But contrast this to the cancer field, you know. In cancer research, people have well-established oncogenes, tumor suppressors, they understand pathways, there are treatments, you know. In certain ways, the theory of cancer is far more developed. There's another lack of consensus, which relates to the first one. If you don't have a theory of aging, you really have a hard time defining biomarkers of aging. So we really don't have any consensus on proxy endpoints of aging and so on. And I contrast this to other medical fields. We clearly have wonderful biomarkers, for example, for anti-hypertensive medications, you know. So, but the key is there's a lack of consensus. And the question is, how do we arrive at consensus in the aging field? And there is a medieval solution, and which was developed by the Catholic Church. So when you want to elect a new pope, you need to arrive at a 2/3 supermajority, which to elect the pope. And at some point, 1271, Gregory X decided that he will lock up the cardinal electors in seclusion and won't allow them to leave until a new pope has been elected. And there's an enforcement mechanism, which is after 3 days, the cardinals receive only 1 dish a day. And after another 5 days, just bread and water. And that has really solved the problem of consensus in the Catholic Church. Now, there was a venture capitalist who thought, I will do the same with the aging field. And so he invited everybody to an island and locked them up. And nothing ever came of it. So this is a picture from this secluded meeting on establishing consensus in the aging field, you know. So what do we do about that? I really think there will be a solution, which is capitalism, you know. So I believe the companies that use effective biomarkers of aging will succeed at developing effective anti-aging interventions. And success will ultimately lead to emulation and then adoption. I want to now talk about some open and closed goals in the aging field. So just some history. What I call old holy grails in the biomarker field. To me personally, I always thought Holy Grail is aging clocks that apply to all tissues and cell types. And why was that a Holy Grail? Because in my worldview, aging happens really in most tissues and cell types, you know. So a biomarker should measure then aging in all of these tissues and cell types. And but then there's another Holy Grail, which is, well, we believe that all mammals age. And therefore, it would be good to have aging clocks that really apply to all mammalian tissues, you know, not just humans, you know. And again, these are pan-mammalian clocks. So, I mean, I think the status here is on these Holy Grails, we have reasonably good solutions based on cytosine methylation. I really think cytosine methylation stands out as a molecular readout because you can accomplish these goals. Contrast that to biomarkers based on transcriptomic data. I mean, this will be rough. This will be very difficult, you know. Develop a transcriptomic clock that works in all human tissues. That's tough. Develop a transcriptomic clock that also works in humans and mice. That's really difficult, you know. But this is, my hope is that the field will find other readouts beyond cytosine methylation, because cytosine methylation is quite limited. You know, we need markers of autophagies, biomarkers surrounding mitochondrial function, stem cell function, senescence markers, all of that, But so I think there are opportunities to address these old goals with new data, perhaps. Now I want to come to what I call new holy grails in the biomarker field. And the first is clearly single cell clocks, right? You have a single cell, one fibroblast, you want to measure its age. And why do we want these clocks? Well, one is that these clocks will allow us to deal with confounding by cell type composition, right? Where bulk tissue is comprised of many different cell types. And we would like to know what's the age of a stem cell, what's the age of a more differentiated cell. But also we need these single cell clocks to enhance our mechanistic understanding of the aging process, coming to the point that we don't have a theory of aging. So maybe these single cell clocks can help us advance that. And so single cell clocks may also explain how molecular changes relate to cellular phenotypes and ultimately tissue dysfunction. And when it comes to single cell clocks, I distinguish 4 different types. The first one is just have a single cell clock that works for one specific cell type. Let it be hepatocytes, or keratinocytes, just for one cell type, solves that problem. You can then probably guess that the next thing will be pan cell type clocks, right? Can you build single cell clocks, but suddenly you apply them to hepatocytes and keratinocytes and endothelial cells? So again, they work in these cell types. Once you've solved that, go to the problem of species. Can you build clocks that work in humans and in mice and in rats, and so on. So I think this is a very exciting area. The status is ongoing. Many research groups work on this topic. For example, last time you saw a very nice talk from Vadim Gladyshev, who presented a single cell clock. But fortunately, many people are aware of the importance of that goal, The other, what I call new holy grail, I was struggling a bit whether I should call it a new holy grail or an unsolved old goal, but it's a causal clock, And so they could fundamentally be based on bulk tissue. They don't have to be based on single cells. The problem with causal clocks is people don't have a good definition of them. So what is meant by a causal clock? Well, several statements. So if you reverse the age estimate of a causal clock, then it will convey health benefits to a patient. Fundamentally, that's what people want, So if you rejuvenate a person by 10 years, you would like that this person will end up living 10 years longer, That would be... And so causal clocks could, of course, serve as biomarkers for monitoring treatment efficacy and ultimately, of course, as proxy endpoint for human clinical trials. And I mentioned mechanistic understanding would be very desirable. Some people would say mechanistic understanding is mandatory. I'm more open-minded about that. To me, I would be happy if somebody uses some artificial intelligence, develops a black box biomarker, and as long as the biomarker meets the above criteria, I would be happy with it, Now, so the question is, where are we with these causal clocks? Do we have them? And now you want to distinguish species, because I would say when it comes to mice, we are almost there. We may, again, coming to the Rumsfeld quote, we already have biomarkers we could use. We have many mouse clocks that respond to anti-aging interventions such as caloric restriction, growth hormone knockouts, and also this parabiosis paradigm that Vadim Gladyshev mentioned in his previous talk. Parabiosis is associated in young blood with lower epigenetic age. And conversely, there's a well-known pro-aging intervention, high-fat diet, obese mice. If you look at their liver, then you will see epigenetic age acceleration. And these interventions I mentioned here have been validated carefully in the field of epigenetic clocks. I would say pretty much any reasonable epigenetic clock will pick that up. Even as I mentioned, my universal clocks pick it up. These are very strong effects. I would say the opposite. If you have a mouse clock and it doesn't respond to these treatments, you have a problem in the definition, perhaps. I also want to discuss rapamycin because this has a lot of interest in the field, and it's well established that rapamycin extends the lifespan of mice. And my verdict is I'm a bit on the fence about it when it comes to epigenetic clock studies. So I've seen data sets and clocks where I saw an effect, and there are even publications on that. But I also saw failures, So just so that with the rapamycin, I would say we need more data. And I know Vadim's just sent me a new data set, so we'll have it soon. Yeah, so coming to the topic of causal clocks, I think it's fair to say in mice, we kind of have it. I'm quite optimistic. And also there several groups do now longitudinal lifespan studies. I would say there will be a check mark under this and within a couple of years. However, we of course want causal clocks for humans. And this is the topic. Do we have human clocks that track anti-aging interventions? And there we face this fundamental problem. We really don't have validated anti-aging interventions in humans. It's a catch-22 situation. Without proxy endpoints, we cannot validate anti-aging interventions in humans, right? Without these biomarkers. Conversely, without anti-aging interventions, we cannot validate the biomarkers, right? You want to test the test. And if we don't have gold standard interventions in humans, we have a problem. And I propose here 3 workarounds. The first is, well, one could actually build some sort of human clock on the basis of genes or proteins that are causally involved in aging, So that goes under the heading detailed mechanistic understanding. If you really have a good understanding of an aging process, you can target the underlying genes and pathways. The second way of dealing with this problem is to actually use non-validated interventions, So we could simply assume that certain interventions reverse human aging. And for example, all of us hopefully exercise, right? We all believe exercise is really good for us. And I would say most people will say exercise rejuvenates you in one way or another. So maybe that's a gold standard intervention. When we come to caloric restriction in humans, we are already getting to murky grounds. Primate studies suggest that there could be an effect, but it's simply not at the same level as rodent studies, right? So, and then many of you, of course, have debated metformin, rapamycin. So one could, in theory, assume that some of these are anti-aging interventions and then use those to build causal clocks in humans. Yeah. There's a third workaround. And this is to leverage these multi-species clocks, okay? So what do I mean? Imagine I have a universal clock and it meets all the criteria in a mouse model. right? All the known interventions that prolong the lifespan of a mouse have an effect on my universal mammalian clock. Then it stands to reason that probably this will also translate to humans. Why? Because it uses conserved cytosines, conserved biology, you know. Again, it's an assumption. Yes. Boy, I already talked for 40 minutes. You need to stop me if you want me to, but here's some, people always ask me these questions. Maybe one more minute, because some of you will ask me these questions. Are epigenetic changes causal, quote unquote, for aging? And I mean, the answer is yes, no doubt about it. Any aging researcher will say, yes, certain epigenetic changes are causal. It's a primary hallmark of aging. But the questions are, but to what extent causal? You know, is epigenetics 25% causal? Is it 75% causal? You know, just to quantitate it, you know, to what extent is it causal? And also in what tissue? Is epigenetic aging in blood causal, or is it epigenetic aging in heart tissue causal? You know, there's a lot of buts, but yeah. And the other thing is, well, how do you define epigenetic change, right? There are histone modifications, non-coding RNA, methylation, you know. So there's a lot to work out, but the good news is the answer, the yes and no answer is yes, you know. But now comes another question, which is a very relevant question. Is cytosine methylation causal? Because everything I showed you today is based on cytosine methylation. And there the answer is definitely ambiguous. So the answer is yes and no. And so one can find plenty of examples really to support either point of view. So recently, David Sinclair and his lab have shown, for example, that TET enzymes are needed for regeneration of this optical nerve. Quote, the beneficial effect of this epigenetic rejuvenation and reprogramming in axon regeneration required these TET enzymes that play a crucial role in demethylating the DNA, you know. So there are powerful experiments that argue for a causal role of methylation. But then there's also very good arguments for no. And so anybody who studies C. elegans will tell you C. elegans doesn't have cytosine methylation, but C. elegans ages, right? So, so if you make the assumption that aging is universally conserved between invertebrates and vertebrates, you have a problem if you say cytosine methylation plays an important role. And also, many cytosines gain methylation or lose methylation with aging, but it really has no effect on neighboring gene expression, you know. So the transcriptomic consequences are often entirely unclear, you know. So that's where we are. Unfortunately, biology is complicated, you know. There's no yes and no. Some changes will be causal, some will not be. And then the challenge for the field is to develop clocks that hone in on these causal changes, right? So I think there, you see some future studies. So we need comprehensive studies between methylation and I say proteomics, especially proteomics or single cell studies. We need clearly interventional studies. For example, you can adapt CRISPR-Cas9 to change cytosine methylation and then study the consequences. You know, that's really a very important frontier to characterize these and address the question. I had some emails with several people on the issue of confounding by cell composition, just to mention it. So cell composition can greatly confound any omics biomarker, transcriptomics, proteomics, and also methylation can be confounded by cell composition. But I want to caution people to don't throw out the baby with the bathwater, because changes in cell composition can very well be part of the signal, okay? Coming to the question, well, what is the cellular manifestation of epigenetic changes? Well, maybe if you change the epigenome, it changes the stem cell functioning. It leads to changes in cell composition. In other words, that could be the part on the causal chain. And this is, in my worldview, again, a selling point of pan-tissue clocks, because overall, they are quite robust to changes in cellular composition. You don't need to obsess about it, you know. But there are many solutions to confounding, and I mentioned them here. I think I'll stop here, you know, and give the audience a chance to ask questions here.
Thank you so much. Steve, actually, can you leave that question up for 1 second? Just in case someone wants to read the final slide about the question. Because I want to ask you one more thing while we're on the record, which is, what can this group or people potentially watching a video can do if they wanted to help? What would be a thing that would be useful, helpful?
I mean, there's so many ways, you know. I mean, I have a non-profit foundation. If you have an exciting study, please contact us, you know, because we are looking for exciting interventional studies. Why? Because they help us characterize clocks, you know, and so that would be, get in touch with that. Many of you work on regulatory aspects, you know, engaging with the FDA, for example. I'm very grateful for that. And yeah, I'll stop with that. Mm-hmm.
Okay, thank you so much. Hey, I'm going to keep my thank yous very, very short because we have a lot of questions in the line, but really thank you so much for joining us and for this very philosophical and hilarious presentation.